Hello, welcome back. I will walk you through the tutorial of protein-protein docking. So you can see in my screen I have here the protocol and the PDF, and on the left I have the terminal. So let's start with the first point. We need to create a directory inside the protein-protein docking directory called my files. So few basic commands to know where you are is ls, and you can see that I'm in the protein-protein docking. I have the input file folder, I have the output file folder, the presentation, and the MD file with the information. What I'm going to do now is create a directory. Make there my files, and then I will enter in the directory with cd. Okay, now we pass to step 2. For step 2, we will need to take an internet page that I prepared here, and I'm already on the website RCSB PDB, that is the protein data bank. You should already know at this point how to download a PDB. Anyway, I will show you again. The first one we have to look is 3GBN. So 3GBN. Here is the structure, and for downloading you have to click here, download file and PDB format. It asks you to save, you say OK. The next one that we have to find is 3GBM, so we do the same things, 3GBM, and here it is. Again, we download file and we download the PDB format. Okay, so now we have to move these two files from the download to our folder. However, we already have the two files in the folder. In this case, I will just copy from the input. So copy from input file. Here are all the input files that there are. We copy 3gbn.pdb here. Let me clear this, and then we copy 3gbm here. So if we now do ls, we can see that the two files are in this folder. Okay, so we are now at this point of the tutorial. Open the two structure in Pymol. If you have Pymol already integrated in your computer, you can type in the terminal Pymol with the two names of the structure. In my case, it is not, so you have to first open the Pymol window, and then I will just drag the file into the main window. Here they are. So what we can see are the two PDB. In green we have 3GBM, in cyan we have 3GBN. We can see that in the structure there are many extra things that we are not really interested. For example, this is a glycan chain, the red cross are water molecule, and this red ligand is probably something that I use for the crystallization and that is present in the structure, but is not really necessary for our modeling. So we will need to clean the structure in order to remove all these, but first let's check the chains. So we can type set sequence view, that is sec_view, comma 1, and here we can see in the top part the sequence of the two protein, or the two PDB. So I will now focus only on 3GBN because we have just a single unit, while for 3GBM we have two. I will color it by chain. So we can see that in green we have chain A, the name of the chain is specified up here. If we scroll to the right, we will find then in cyan chain B, and this is the epitope of influenza. hemagglutinin. Then we have in pink and in yellow the antibodies, so H is the heavy chain and L is the light chain, and here they are. You can do the same kind of analysis for 3GBM. The next thing we want to do is to align the two structures, so you can type align the first partner, that is 3GBN, comma the second partner, that is 3GBM. So as you can see now, they are aligned. I will color them into different colors, so we do 3GBM in a lime green and we do 3GBN in a blue. So here you can see the influenza epitope overlap quite well, especially in this area. The antibody also is quite nicely overlapped, also if slightly displaced, but this we know because they are two different epitopes. So what we will need is basically the hemagglutinin chain A and B from 3GBM, so the green structure, and we want the antibody structure from 3GBN. And this is what we are going to do next. Okay, so now we want to clean the structure, and we are in section 2. So for the PDB structure 3GBM, we want to extract only chain A and chain B. And we want at the same time to isolate this two chain and also to remove all the extra atoms that are not needed, like the water molecule that we saw, the glycan, and so on. So cleanpdb.py is a Python script that was developed in the lab in order to remove all the extra residues that are not protein related from the PDB. And what you need to do when you use cleanpdb, you recall the script and then you put here the name of the PDB that you want to clean and the chain. So in this case we have A and B. If you have 4 chains that you want to isolate, you put the 4 letter one after the other. If you have just one, it will work. Anyway, so first things we need to call Python 2.7 because this script was written in Python 2.7, and if you use version 3 it will not work. So this is the reason why you were also asked to install Python 2.7. Then you have to type the path to the Rosetta folder that you have. Mine will be different from yours, so you have to kind of discover what is your path. Mine is doors, Meiler Lab, app, Rosetta 3.12, because this is the version that we are going to use, and then we can just copy what is present here in the PDB, in the tutorial. So here it is. We have here the first FASTA sequence. We have it printed in the terminal, but this will also be present in the folder. This is the second chain, chain B, and let me clear. If we check what there is in the folder right now, we can see 3gbm.pdb and 3gbn.pdb were the two files that we had before. Then we have the two FASTA sequence for 3gbm, chain A and chain B, and then here we have the PDB that should be cleaned and isolate chain A and B. So the next step that we want to do is to renumber the chain. Afterwards I will open the PyMol session and show you what I mean, but this structure is not starting from residue number 1, and we want to renumber it in order that everything starts from number 1 and go on in a linear way. So the next command is PDB renumber, that will take the PDB and start from amino acid number 1 and go on until the end of the protein, or the proteins in our case. I will not type again all the paths. You will probably have to do it. Anyway, I already prepared the command and I will just paste it here for being more rapid. So now if we check what we have in the folder, we still have the two initial PDB. We have the FASTA and the FASTA that we obtained before. This is the PDB that is not renumbered, is just cleaned, and here is the renumbered version. So before showing you in PyMol, I will do also the other protein PDB, so I will extract in that case the antibody chain H and chain L. So again we need to call Python and clean PDB, and in this case we are recalling for the 3GBN PDB structure and for the chain HL. And again I will just paste it because I want to save some time. So here you have the FASTA, and if we now check what we have in the folder, we have the initial 3GBN, we have the FASTA sequence for both chain A and B of 3GBM and for H and L for 3GBN. We have the cleaned but not renumbered structure for 3GBM, the renumbered structure, and then here we have the PDB of the clean antibody. Okay, so in the next session I will show you in PyMol all the different files we created now. Okay, so now we visualize the structures. Here they are, I already loaded in PDB. So if we check the sequence, here we have the influenza hemagglutinin protein, chain A in green, chain B in cyan. As you can see here in the sequence, we are starting from amino acid 1. We are going all the way back to the end. This will be residues 324, and then we start with 325 for chain B. Now, for the antibody, here are the two chains. Oops. So there are a couple of things that we need to fix in the antibody. For example, you can see that here there is a missing loop because probably the structure was not good resolution for this portion. We have many chain break both in this section and also in this section, and we have to do two things. First, we don't really care about this portion of the protein because this is not the one that is binding the antibody. The one that is binding the epitope. The one that is binding the epitope is this region here where we have the CDR loops, and this portion of the protein is not really necessary. We want to remove it, one, because it's very low resolution, we have missing residues and break chains, but two, because we want to make the calculation time faster. So the smaller the protein, the better it is. So for doing so, we first have to select the amino acid that we want to delete, and for doing that we type in the PyMol terminal sele for selection, then we give a name to the selection that we want to remove, in this case to delete. We put a comma, and then we identify the residue, so we do res e, that is index, and we give the range that we want to remove. So the first range is 121 to 160, and I can show you which residues are. So 121 to 160 is all this portion that we don't really need, and the second section we can add it with a plus, and the range is from 268 to 311, and is basically from 68 to 11. So we are removing also this portion here at the beginning. We click, we create this selection that is exactly what we already selected before, and now to remove it we type remove and the name of the selection, that is to delete, and that is gone. So now we want also to save this structure, and for doing we do save. We give a name for which we want to call it, that will be 3GBN trim .pdb, and we recall the structure that we want to save, that is 3GBN HL, not the other one. So we give 3GBN HL. And if we now go to check our folder, here it is. So the last things I want to show you Before I mentioned that we need to renumber the PDB, and now it's really essential because, for example, we remove all these C-terminal portions. So here we have a jump from 119, 16. Sorry, this is 120 and this is 161. So we want to renumber it in order to have a continuous numbering scheme. The reason why we want to renumber is that Rosetta itself will consider the numbering of the pose. So if you are removing a residue in here, Rosetta will not consider the numbering of the PDB. So in this case, the residue V 161 in the PDB, Rosetta will read it as V120. So for this reason, to make easier to the user to recognize and to recall the correct amino acid, we will renumber. So this is the next section, and what we will do also next is closing this break chain that you can see here, because this is a portion of the antibody that we will need to use, and so we want to make sure that this section is fixed. Okay. So now we just saved the trim version of the antibody. We can see here in the folder. The next step is renumbering, and I just realized that there is a typo in the PDF tutorial because here should be a double dash as we had here. So if I will not correct it in your version of the PDF tutorial, pay attention, you have to write a double dash. Anyway, I have the command here, so I will just copy and paste, and now if we do ls, we have this PDB that is the renumbered version of the trimmed antibody. Good. So now we pass to the third section. So we close the chain break that is now between serine 127 and valine 128. The first part of this section is open the PDB and visualize it in PyMol. I will skip it for a time reason, but you can be free to open it and check how the renumbering changed the residue numbering identification. So I will directly skip to this command, and what we want to do is open a text file that is here. For me is already ready. You can also copy from the input structure or from the input files, and this is a file that describes or that instructs Rosetta to build a loop between residue 125 and residue 130. So we have a couple of residues spanning on both sides of the break chain because we want to give enough freedom to build a loop in that position. Okay, now before starting the chain break closure, we just need to copy the options. So we are here. We will copy this in our folder. If we now check what we have in here, we have both the chain break fix loop we created before and the option file, so we are ready to launch the run. We will ask 10 output structure, and it will take more or less 10 minutes, so I will stop the video and come back with the result. Here is the command, and as you can see it's running, and I will be back when it's finished. Okay, the chain break mover finished, so now we can check what it did. I open already a PyMol session with all the 10 structures that were created, and you can see that for the majority of the antibody nothing changed, but here where there was the chain break, we now have 10 models that are fixing it. So we will now check the score file that we obtained for this run, and we want basically to extract the best one. So let's do like this. This is very messy. We can maybe do like this. So the second column is the one indicating the total score, and we sort them in order to have the lowest number first. And so our model, that is the winning one, is number 07. So this may differ for you. Just check. In the tutorial we say that was number 2, but it's not in this case. Anyway, we are now ready for the next section. First, we will want to copy the antibody that is the best scoring model in our folder, and we want to rename it 3GBN AB fixed. So in this case, since my model was 07, I will just rename that, and I will copy 3GBN AB 007.pdb, and I will rename it 3GBN AB fixed.pdb. So if now check in the folder, we have here all the 10 models, and this is the one that we just create based on this. You can still take the best one that is in the folder, the number 2. It will not change so much, but when you will work on your own, and especially if you have a break chain that is much bigger than what we had, maybe you want to pay attention to take the lowest energy. Okay, so we just closed the chain break and we visualize it on PyMol, and now we have to repack the structure. The reason why we have to repack the structure is that closing the chain break may have inserted some small clashes, and we want to avoid to have them during the docking run. So we have to repack the structure of the antibody that we just selected as the best one according to the Rosetta score after the chain break closure. And 2, we want also to repack the antigen and the epitope structure, so I don't remember the name, 3GBMHA, so the influenza protein, because then we will have to combine them together in order to start the docking, and we want that the structure are pre-packed because we give a better energy and will be easier to manage afterwards. So I will now copy the file that we need. We have repack.xml that will be needed for repacking the antibody, and this is a repack that will act on all the residues, and all the sidechain will be repacked. For the epitope for the influenza protein, we will use a slightly different protocol that is repack HA, and in this case we select 3 residues that we want to prevent from repacking, because we know that these residues are important in that exact conformation for the binding. So the 2 protocols are different. We will run one after the other. I will show you only the first one, and then I will come back for the point number 4, but you need to do both steps. So first things, let's copy all the input. Ta-da! And here we have protocol with restriction, protocol without restriction, and option for both of them. And now I will launch the first one and the second, and I will come back later. So see you soon. Okay, so the 2 repacking run finished. We have one in here and the second in here, and we can see in doing ls that we create 2 new files that are 3GBM HA001.pdb and the 3GBN AB fixed. So when you do repack in your system, you may want to create more than just one output. So you want to create at least 10, for example, and then select the best scoring one. For a time reason, here we create just one, and we will use this as input for the next step. So we will just rename them as described here. So we will copy from the output file Actually, we create more repack structures, so you can find more than just one, and those are better score compared to what we create here. So that's all, we copy them, ls, and we can find the repack 3GBN antibody and the repacked epitope. Okay, so the next step that we will need to do is to orient the antibody in the proper starting conformation compared to the epitope. And once that is done, then we are ready for starting the docking. Okay, so we are almost ready to start the docking. The last step we have to do in the input preparation is to orient the antibody in front of the binding site on the epitope. And for doing so, we will use the structure 3GBM native that is in the input file. This structure was pre-prepared by us for you so that you save some minutes, and is basically taken from the 3GBM PDB that we download from the protein data bank, and we extract all the chain A and B, so the epitope, and also H and L, the antibody. And what we will do, we will use this as template in order to orient the antibody of the other structure together with the epitope of 3GBM. So I already opened the PyMol session. Here we can see the native is basically what I was saying. We have all the 4 chains. Here there are the 2 chains of the antibody, and here the 2 chains of the epitope. And then we have 3GBMHA, so the epitope that is here. And as you can see, it overlaps exactly the native, but the antibody is far in space. So what we are going to do now is to take the antibody here and align it to the native structure, so to the green antibody, in order to position them in the correct initial position for the docking. So for doing so, we will type in the command line align, then we will call 3GBN repacked and comma, and then we will call 3GBM native. And that's all. So you see that now we have everything aligned in the correct position. You can see that the antibody doesn't overlap perfectly because these are 2 different crystal structures. So we can remove the native now, and you can see that the 2 are in the good position for starting the docking. We save it, calling it 3GBMHA with 3GBNAB, and these are the 2 chains that we are going to concatenate basically in the PDB. So let's do it. And the next thing we will need to do is to renumber everything, because these 2 will have different numbering. Probably this one will restart from 1, so we will need to renumber everything again as we did before. And I will quickly do it in here before starting the docking. So if now we do ls, here they are. Oh, and my dog arrived. Okay, so we are officially ready to start the actual docking. As I mentioned in the presentation, preparing the input is the longest part, because you have to make sure that everything is correct to minimize error or problems. So now we have everything ready, and we can start the dock. We will need to copy the XML protocol, so here it is. We will need to copy the option file for the docking, and here it is. And I will not go with you through the XML. I already presented in the presentation with the slides. You are free to familiarize yourself if you want. So me, I will step directly to step 3. Here we are creating 10 structure, and it will take more or less 5 hours. So each structure, each single model will be produced in more or less 30 minutes. And for reason of time, we just ask 10 structure, but be free if you are working on your own subject to increase this number. The minimum you should do is 500 structure. the ideal is 1000. And this for the local docking. If you want to try the global docking, where you have no idea where the binding site is, you may want to increase this output number to up to 5000 or 10,000. Okay, so let's go. I will start the run. Oops, here I didn't copy yet. And now let's check the file. We have docking XML and docking option, and we are ready to start. And here it is. It will run for 5 hours. I will let it go overnight, and tomorrow morning I will finish the protocol. Okay, good luck. Okay, welcome back. We ran the docking protocol overnight, and we are ready to go on with the next step, that will be analyzing the result and preparing the input structure for comparison. So if you don't want to wait 5 hours of the run, you can easily check output, the folder output files. We created more than 10 docking runs, so you can just copy that into your folder. And let's go on with the next part of the protocol. So let's first check if we have all our model. So as you can see here, these are the docking run. We have missing 0001 and 0010, and this is something that I told you it may happen, because if the run fail, Rosetta will not produce the structure and will just pass to the next model. And now, before plotting the result and analyzing both the total energy and the binding energy, we want to minimize the input structure. And in particular, I want to minimize our control, that is the native, because it's the one that we will need for comparison of the result. So we now pass to this portion. We need to copy an XML file that was designed in order to minimize the native structure. And we copy the XML, and then we run the minimization on the native structure. So it will take more or less 1 hour. I will be back when it is finished. Again, if you want to save time, you can still go in the output file and copy the protein that we will need for the final step. Okay, that is running. See you later. Okay, so we just finished to minimize the native structure to use as control for the total energy and binding energy of our model. And we can check the file doing ls. And here there are the two, minimize 1 and minimize 2. So, okay, we are ready for the next step. So in the next step, we are going to use the mover interface analyzer in order to extract the information about the binding energy of our models that were docked. For doing this, we need to copy two files, the XML and the option file for the interface mover. Be free to open the XML and see what it says. So first we copy the file, we check we have them, docking analyzing option and XML, here they are. Okay, so now we are ready to launch the analysis. And here we are basically calling all the input that we create with the docking run, so the full and also the minimize, and it will take some minutes to calculate, and then we will analyze and plot the result. Okay, so before checking the result, we analyze the PDB structure that we created. So I opened them in PyMol. These here are the models that were created by the docking run, and this is the native. So first thing, we will align everything to the native in order to see how good the analysis and the docking was. So as you can see, the alignment is not perfect. We can color everything in gray and the native in red, so it will be more clear to see. And we can see that the binding is not perfect for all the models, so this is slightly away. We have also this that is very distorted, and so on. So in theory, the lowest RMSD and lowest energy structure should be the more close to the native. So here we have only 10 structure. In the output file we have many more structure, and we will then check which one is the lowest energy and compare the two of them. Okay, let's go to see if the interface analyzer finished. Okay, so now the interface analyzer mover finished. We obtain all the information for the binding energy, the total energy, and also other information that we can extract from the file. We should have this new file called dockinganalysis.csv. That is a table that contains all of them. And since you prepare only 10 structures, this is not really optimal to plot. We usually will produce at least 500, but for reason of time we didn't in this occasion. But you can copy from the output file folder the same file in which we pre-created more runs, and you will have more model for preparing a nicer plot. So let's just copy dockinganalysis.csv from the output folder, and now we can display it on the screen. Will be a little bit messy. Here it is. So you can see that we have much more structures, and here in the second line you can find all the information that are contained in the file that were calculated. So we will be interested in the total score that is in column 2, and we will be interested in DG separated that indicate the binding energy at the interface. So you also have other information, so the SASA, how much change the exposed surface, for example, and so on. You be free to open the CSV file and check out also the other parameters. We will just check the total score and the DG separated. Now, oops. Okay, sorry. So now for plotting, we have two ways. You can open the CSV file in Excel, for example, and create the plot from Excel. We don't really recommend Excel because if you have tons of data, like if you really do 500 or 1000 data, Excel will be really slow. You should learn some basics in R, so we already add some script that you can use if you want, so you can basically run these. Now I already have prepared the graph, so I will show you directly. We have here the total score. Can I have a little bit of zoom? Okay, here it is. So in pink there are all the models that are more than 10, as you can see, that were docked, and in blue we have the native structure minimized. So you can see that we have a kind of funnel plot like. Now imagine if you have 500 structure, this area will be much more populated. Anyway, we are happy to have reached at least the same, if not surpass the energy of the native. Now this protocol was really quick, was a quick tutorial, and we only have 2 minimized structure for the native. But if we had 10, for example, it can be that we didn't really reach because one of the 10 could have been down here. So this regarding the RMSD versus total energy, and here there is the RMSD versus binding energy. So this is really the energy at the interface. Again, we have a funnel-like plot. If we had 500 structure, this area was much more populated. And in blue we have the minimized native structure. you can see that the binding energy for the two minimized is very similar, while for the total energy it was different. So it means that the interface is very conserved here, so they maintain almost the same interaction and the same energy in the binding. However, the total energy of the full protein changed in the total energy that we saw before, and this can be simply due maybe a loop that is misplaced in one structure and not in the other, so a hydrogen bond, for example, is formed or removed. Okay, anyway, the docking run works very nicely because we obtain models that are better than the native. So we are going to check that one in PyMol, and we know that is number 35 in the output. So let me copy. From output file 35 here, and now we will check on PyMol these two structures. Okay, so I already opened in PyMol the two structure. We have in green the best model that we obtain according to the binding energy, that was the number 35. In cyan we have the native, so we first align them. And we can see that the alignment is very nice, so the two structures overlap really well, especially here in this area. They are not perfect, 100%, but we also created less than 100 docking models. So if you increase the number, you are probably more close to having really the native structure. Anyway, the results are really good, and I hope you enjoyed this tutorial about protein-protein docking. If you have any question, don't hesitate to write me or to write in the chat, and we will be happy to answer your question. So, good luck, and let me know if you have questions. Bye!